to chapter 14. So we're going to do this in one part. Really not a bad chapter, really a lot of definitions, a lot of more memorization stuff. So we're kind of out of all the big concept chapters from exam 2. So anyway, we're going to start out with what epidemiology is, and we're really dealing with diseases and how they are transmitted, when, where, all that stuff, and how. We look at things over time. We look at vectors. We look at things like the people, and there's ways we can try to understand it with spatial analysis, a variety of other things that go on. Pathology is study of disease. The pathogen is the microbe that actually causes the disease. And then we have etology, and that's the study of the cause of disease, what's going on at the cellular level kind of stuff. Pathogenesis is development of disease. Infection is actually getting a pathogen in, colonized by a pathogen. Bacteria gets into the skin, gets into a mucous membrane. Virus gets into the body. Disease is a state in which the body does not function normally. Many diseases are caused by microbes. Remember, that is the germ theory of disease, is that many diseases are caused by microbes, except things like cancer and genetic diseases, things like that. So it's a change in the state of health of a person that we can see. Now, there are some things I like about, like morbidity. That's actually the incidence of disease. So how many people have the disease? What's the morbidity? Morbidity is how many people have it. Mortality is how many people die. We have what's called notifiable infectious disease. These are things like people have to report to the health department. Early warnings for outbreaks, things like smallpox or something. Okay. The World Health Organization is our global surveillance against diseases. And we also deal with things like virulence, and it's like how, to what degree is it pathogenicity of it is, to what degree, how easy is it to spread, kind of thing. Okay. Now, when we talk about the normal microbiota, you have about 1 times 10 to the 13th power body cells, versus bacterial cells is 1 times 10 to the 14th power factor 10. There are more of them than there are of us. There are actually more bacteria in your body than there are your own cells, okay, than there are human cells. So that's pretty important. The normal microbiota that colonize the human body but don't produce disease under any kind of normal conditions. There are parts of our normal microbiota that can cause disease when things get abnormal, but when things are under normal conditions, they're just there. They don't hurt us. They oftentimes are just living with us. A transient might be there for several days, even months, and they disappear. Transient microbiota, something we got temporarily. Okay. Give me 1 second. I'm sorry. I was just making sure everything was okay. My phone kept going off. Okay, so it was distracting, so I was turning everything off. All right. So anyway, so we're going to deal with that. So some microbiota that we have that live in our nasal cavity, our stomach, our large intestines. There's normal microbiota occupying these spaces, occupying these surfaces, and that in itself is a very big thing the body does. Your large intestine, for example, has the largest number of normal microbiota that we have, though you find it in your upper respiratory system. You find it in your eyes and mouth, your skin. Large intestine is by far the largest. It's in our reproductive tracts, our urethra, vagina in females, urethra males and females, things like that. Normal microbiota, one of the things they can do is they can basically live in places where other bacteria could live, occupying spaces. A competitive exclusion, that's one of the ways they can do it, is our book doesn't go into. They just happen to occupy space, but they battle for nutrients, they produce harmful substances, antibiotics, and they also change the local pH, preventing some things from growing. So there's a bunch of things that normal microbiota do to benefit us to prevent the growth of things, for example, like C. diff. Okay? Symbiosis, when things live together, human and the microbiota. We got what's called commensalism, mutualism, and parasitism. Commensalism, you live together, you don't hurt each other. Mutualism, you live together and you benefit each other. Parasitism, one harms the other. Okay. Commensalism is when one organism is benefits but one's unaffected. So, for example, I live on you, I benefit, I don't hurt you, but I don't help you. Okay. Like the cornea bacteria that live on our eye. Mutualism is both organisms benefit. E. coli in the large intestine, they get home, they get stuff, they get food, we get vitamins and stuff from them. Probiotics, these are microbial cultures that have a benefit when they are ingested for bacteria. Live microbial cultures applied to or ingested to exert a beneficial effect. So this would be like you drink a kefir, however people like to pronounce it, the kombucha, things like that. Prebiotics are chemicals that promote the growth of probiotics. So there might be prebiotics in the drink as well as probiotics, or just one. Parasitism is one thing benefits, the other is harmed. Nutrients extracted from a host, good example is gonorrhea, a lot of your worm infections, things like that. Opportunistic pathogens, they don't cause disease normally, but they will, like in a normal habitat of a healthy person, but they may do in a different environment. E. coli is harmless in your large intestines, but if it shows up in places like your urinary tract, your lungs, your spinal cord, or wound, it can really cause problems. In urinary bladder, it can cause bladder infections. In lungs, it can cause a pulmonary infection. In the spinal cord, it can result in meningitis. In wounds, it can cause abscesses. So there's lots of things that go on with pathogens. For example, E. coli, normal and happy and healthy in your intestines, not healthy and healthy anywhere else. So that's why females have to learn to wipe properly front to back, not back to front, so you don't get fecal matter in your urethra. There is a guy, Robert Koch, whose picture was on the front slide. Robert Koch, he was the one that showed microbes actually cause disease. He determined which we can get the etiology. He used to prove that what was the cause of an infectious disease, which pathogen, and he used what's called Koch's postulates to do so. We know that some pathogens can cause several conditions. For example, the Yersinia pestis can cause bubonic plague, pneumonic plague, and septicemia plague. Some pathogens cause disease only in humans. Some can cause disease only in dogs, for example. So some diseases only affect certain hosts. Some diseases can affect multiple hosts. So, for example, the anthrax disease, he showed that it was Bacillus anthracus that caused that, and then M. tuberculosis caused tuberculosis disease. Okay, so Koch's postulates, he said these 4 things here. Okay, and I will test you on what Koch's postulates say now, and say which of these is. There will be a question about Koch's postulates on the test. Koch's postulates is like, so if you have a disease, this pathogen must be present in every case of the disease. Okay, if you have tuberculosis, then M. tuberculosis must be present. If you have anthrax, then Bacillus anthracus must be present. Okay, in every case of that disease. So that's one thing that shows, okay, this is the pathogen behind it. The pathogen must be isolated from the disease host and grown in a pure culture. So we got to be able to get it, swab you, get it, put it on a plate, and say, okay, we grew this bacteria from the person who had the disease. The pathogen from pure culture must cause disease when inoculated into a susceptible lab animal is healthy. So if we give it to a lab rat and then they get sick, okay. And the pathogen also must be isolated from the inoculated animal and must be shown to be the same pathogen or the original pathogen. So these are Koch's postulates to test for that this is in fact the pathogen that makes this disease occur. Okay. So we have a healthy rat, we get some red blood cells, causing agent is brought, we inject it with it, it dies, it gets sick. We get a disease organism, we get the suspected agent, we culture it from it, we put in something new, we actually get the culture agent from the dead animal too. We find out, okay, let's say we go to the woodland and we find a disease that's making rats sick, rodents sick, and killing them. And we test it, and we do that, and we figure, okay, this is the pathogen that's doing it, this is the pathogen involved, now we can go about how to treat said pathogen, okay? Now, when you get infectious diseases, you're going to get signs and symptoms, okay? A symptom is a change in body function because of the disease. May not necessarily be apparent to an observer, okay? Like pain or malaise. You ask the patient how they feel, and they tell you. A sign is a change in body can be measured or observed as a result of disease, like a lesion, a pustule, a bubo, a swelling, a fever. They're paralyzed. They're having a flop sweat, that kind of stuff. A syndrome is I have one cause, okay, but multiple symptoms. A specific group of signs and symptoms accompanied by a disease. So one cause results in multiple symptoms. Let's just say one cause, not one causes. I'm going to go ahead and just say that on my original, because if I don't do it, I'll forget. Alright, anyway. Let's talk about communicable diseases, contagious diseases, and non-communicable. Communicable diseases spread from one host to another. For example, chickenpox was easily spread. When I was a kid, before the chickenpox vaccine was a thing, my mother wanted me to get chickenpox when I was young, not get it when I was older. So what happened is my cousin got chickenpox in school. My mom took me over to go play with him. I got chickenpox. I got it really bad though. Now measles is another one. Genital herpes, tuberculosis, for example. These are all quite communicable diseases. Some viruses really easy to spread, some harder to spread, some only spread through certain kinds of contact, like sexual contact, things like that. Some you have to get water droplets, you got to get respiratory droplets. Contagious disease is easily spread from one host to another, like chickenpox and measles. Measles has a really high R0, really easily spread. Non-communicable diseases, they're not transmitted from host to host. They are usually picked out from outside the body, from the normal microbiota, like soil bacteria, like Clostridium tetani. You step on a nail, Clostridium tetani is on the nail because it was in the soil. And it was on a rusty nail that you stepped on outside. It was in the soil. It got on the, like rain made it get onto the nail. You stepped on the nail. It gets in the musculature. Now you have tetanus, and you'll have the signs and symptoms of tetanus. Occurrence of disease, there's incidence, prevalence, and then there's what we call sporadic disease. Incidence is the fraction of the population that contracts the disease in a specific time frame. Okay, it's an indicator of spread. We've been studying AIDS since the 1990s, for example. Prevalence is a fraction of a population having a disease at a given time, regardless of when it actually appeared. Okay, so fraction of the population that contracts the disease during a time, and then prevalence is how many have it. Okay, how many have it? It doesn't matter when it appeared, how many actually have the disease currently. Sporadic disease occurs occasionally in the population, like typhoid fever or salmonella. They might be things that show up during barbecue season or summer season, things like that, okay, where we might see these particular diseases go up because people are eating, having barbecues in the summertime, and it might go up more because food's sitting outside with mayonnaise products in it, things like that. So AIDS cases, AIDS was starting to show up in the right about when I was born. I was born in 1982, showed up a little earlier, but I was born in the early 80s. So during the 90s, late 80s, early 90s, it was really bad. Mid 90s, it was really bad. One of the things that actually happened was we started looking more and we found it more. That is one of the things that you will see about disease. When we expand surveillance and case definition of the disease, and we start saying this is the disease, this is what we're looking for, we need to start looking for it, investigate it, and actually surveil for it, and test for it, and expand our testing, we will start seeing more cases. Cases will go up. We will see more incidents as our ability to detect the disease goes up. Our ability to find it goes up. Our tools get better. Okay. So anyway, you can see it took a long time to get to the first 250,000 cases. Didn't take very long to get to the second 250,000 cases at all. Okay, with tremendous expansion in HIV. There's diseases that are endemic. Endemic, this means they're constantly present in a population, like the common cold. There's epidemic diseases. They many hosts in a given area in a short time. Things like influenza becomes epidemic. COVID, was pandemic, a worldwide epidemic. STDs can become epidemic. Pandemic is a worldwide epidemic. Things like AIDS, influenza becomes pandemic. COVID was the pandemic. Spanish flu, Spanish influenza. And how fast things does depends on the immunity of the population. So is there herd immunity? Is there people, have they been vaccinated and have gotten the disease and fought it off and become immune that way too, or both? Okay. Is there natural immunity? Is there vaccine-mediated immunity? Is there some kind of immunity in the population? Herd immunity, we got to have a good chunk of the population vaccinated in order for it to work. Now, acute disease, because that's that herd immunity thing we were talking about there, is because of herd immunity, we have to have a high number. Now, acute disease, they have symptoms develop very rapidly, only last for a short time, like the flu. Now, you will have symptoms of the flu for a while after. For example, the cough may persist for a long time. But the disease is only a matter of days, like influenza or cold. Chronic diseases develop very slowly, continue for long periods, like mono, Hep B, and tuberculosis. Subacute diseases, symptoms actually are between acute and chronic. A rare brain disease called sclerosing panencephalitis is subacute. It goes back and forth. Latent disease, there's a period with no symptoms, but the virus is inactive. It's in a dormant state, like shingles. The virus is actually dormant in the cells. There's other things that cause disease, like predisposing factors that make you more susceptible to disease. Things like, for example, that we now know is having diabetes and things like that. But there's other ones I want to talk about. A short urethra in females makes UTI infections more prevalent. Males have longer urethras because the urethra passes through the penis, and also men, the penis is separate from the you have the vulva with the urethra, vagina, and all in there nearby the rectum. Inherited traits like a sickle cell gene can make you immune to malaria, give you resistance to malaria. Climate and weather can actually be a factor. Like, do you happen to live in a climate that allows for that particular thing, like a mosquito-borne virus? Do the mosquitoes actually replicate enough can they transmit the virus? Is it tropical or subtropical? Are you fatigued? Other environmental factors that can play a role in being predisposing. Humidity, being wet all the time, things like that. Nutrition, how well are you eating? Are you eating a proper, all the nutrients you need? How old are you? Being old. Your occupation can put you in various predisposing factors. Being a medical provider during a pandemic, for example. Pre-existing illnesses like high blood pressure, cardiovascular disease, diabetes, that kind of stuff. COPD, emphysema. Your lifestyle, like having unprotected sex and sharing needles, and also being on chemotherapy. Now, we're going to talk about the stages of disease. When you go through a disease, diseases have what's called an incubation period. And that's, I got infected, and then the disease has got to grow in the body. It's got to replicate, whether it's bacterial or viral. It's got to replicate till it finally gets enough of it to actually cause symptoms. Okay? And these symptoms start to show up very early on. It's early mild symptoms of the disease, called the prodromal period. It's short. Then there's a period of illness where you actually have the most severe disease. It's where the signs and symptoms are really overt. This is where you will have, for example, the really swollen lymph nodes, a sore throat, diarrhea, vomiting, fever, chills, aches, cough, all the kinds of things that you will see. And if the patient doesn't overcome their symptoms, the patient will die if they can't overcome their symptoms during this period of illness. There's a period of decline where the signs and symptoms start to subside. Then there's the convalescence, where the patient's body strength returns to normal, their pre-disease state, recovery's occurred, they can still spread it. They still are infectious, but they are feeling better. So they're, depending on the disease, they can feel better but still spread it. And you have to be careful with the disease and look up during that convalescence period when you still infectious. So there's the incubation period, which is pretty short. The prodromal period, which is also pretty short, mild signs and symptoms. The period of the illness where you're actually the most severe. Then you still have signs and symptoms, but it's in decline. And then you're in recovery, and it's this nice bell-shaped curve. Okay. There are lots of ways we can spread infections. There's reservoirs of infections where it's a continual source of infection. Could be a human, an animal, or a non-living thing. For example, human carriers may have no signs of illnesses like HIV. They may have the virus but haven't started to show the disease. Gonorrhea, hepatitis, amoebic dysentery, and you can get that from them. Animals that have zoonoses, they can be transmitted to humans, like rabies is one of the really nasty ones. Rabies is diabolical, terrifying virus. Lyme disease, for example, can be spread from animals. That could be non-living, it could be in the soil, it could be water or food. Okay, botulism, tetanus, ringworm. Tetanus is a soil bacteria. Ringworm is a fungal. We'll talk about botulism later. Now, there's loads of ways to get transmission diseases by contact. There's direct contact, like physical contact, person to person, touching, kissing, and sexual intercourse. This is things like getting influenza, staph bacteria, various STD, STIs, and mononucleosis. There's indirects, like use of a non-living object, like fomites, like touch. And a fomite is basically, what I like to think of a fomite is something that is a place that bacteria and viruses are on, but it's easy to pick up and take with you. This is a great example of a fomite. It's our smartphones, but I usually think of it as something you can easily pick up and carry with you. But some people might be like your fomites. I think anything else is surfaces, but like your grocery carts, things like that can have stuff on it, towels, bedding, cups, money, eating utensils, things like that. Indirect, from touching a shopping cart. Remember when we all were taking our food and bringing it home and wiping it down with Clorox wipes and everything during the pandemic. Droplet transmission. Microbes can spread droplet nuclei, mucus droplets. They can travel short distances from coughing or sneezing or even talking. This is a really good way of spreading influenza, pneumonia, and whooping cough called pertussis. So direct contact, this can be just within friends, very platonic, like grabbing each other's hands platonically, or sexual contact. One of the ways that we direct contact through surgical surfaces, indirect contact transmission, cups, cleaning up somebody's snotty rags, droplet when we sneeze. There's vehicle transmission through water, food, air, blood, other body fluids, drugs, and IV fluids. If it's waterborne, they're contaminated or untreated or poorly treated water that has sewage in it, like cholera. People drink dirty water and get cholera. If you played Red Dead Redemption 2, that's what killed the town of Armadillo, was cholera. Shingillosis. Foodborne, incompletely cooked, poorly refrigerated, prepared under unsafe conditions that cause food poisoning or tapeworms. Airborne, the respiratory droplets travel more than a meter. So if somebody sneezes in one aisle, it spreads to the next aisle. This is how tuberculosis spreads. Measles is respiratory. Dust particles, things like that. Okay. So water, food, and air are all great ways in which we can transport pathogens. You get Giardia lamblia by drinking water. There's other vectors. We'll talk about vectors. Many of them are insects, but these are animals that carry pathogens from one host to another. Arthropods are the most important groups. These especially are ticks and mosquitoes and other biting flies, like the trypanosoma by the African sleeping sickness, by the tsetse fly. Mosquitoes, other biting flies can transmit diseases. Mosquitoes and ticks are great examples. There's mechanical transmission, just insects' feet or other body parts. The insect walked on some material that was infected and transmitted it. Biological transmitted from insect bites of an affected animal, like the saliva carrying Plasmodium. Pathogens actually reproduce inside the vector. They can be transmitted to another host and can be passed with feces, vomit, or saliva. So that's kind of one of the ways that mosquitoes can transmit the Plasmodium that causes malaria. Nosocomial infections are hospital acquired. These are things you get in the hospital. 5 to 15% of all hospital patients will be affected by a nosocomial infection. These are things like MRSA, for example, that you get staph infections, nosocomial. Okay. What can cause nosocomial effect? The microbes in the hospital. We'll see those on the next slide. Some ones we'll see. But a compromised host is infections impaired by disease because they are sick. That's why they're in a hospital. Chemotherapy, for example, or other kinds of therapies, or they're on burn. They have burns. And broken skin or mucous membranes and a suppressed immune system, like T and B cells, are suppressed. There are two principal routes we call chain of transmission, two principal routes: direct contact from staff or patient to patient, or indirect through fomites or hospital systems, hospital ventilation. Like they were putting a tube in and it was infected, some kind of fomite, tools, things like that, had something on it. It was in the, or it went through the ventilation system because there's staph on the walls and stuff like that. There's coagulase negative staphylococci, 25%, 89% of them are actually resistant to antibiotics. Or Staph aureus, 16% of total infections out in the hospital, 80% are resistant to antibiotics. Enterococcus, 10% of infections, 29% are actually resistant to antibiotics. A lot of gram-negative rods, that's a lot of your fecal coliforms. 23% percentage of infections, so another high percent, but 5 to 32% are resistant to antibiotics. It just depends, highly variable. We don't know of any here with C. difficile, but C. diff is very hard to treat. 13% of total infections is C. diff. So coagulase negative staphylococcus is a big one. Okay, we'll see a lot of them are urinary tract infections. Next, we have surgical sites. Then we have some other things like respiratory and bloodstream infections. And then we just have other, the second most. Emerging infectious diseases are new or changing diseases that increase potential to cause disease incidence. I said increase in incidence. Sorry, I've been recording today, so I'm getting pretty tired already. So there's EID criteria that identify them. They have clearly distinct symptoms. It's a new pathogen. It's a local disease, but it's become widespread. A rare disease is more common, or a mild disease becomes more severe. That's kind of what happened with COVID is emerging infectious disease. Okay. Genetic recombination. There's new strains of E. coli. Those O157. There's avian influenza H5N1. Evolution of new strains can happen with mutations in DNA replication, like what happened with Vibrio cholera O139. I always have a hard time remembering those numbers, to be honest with you. Inappropriate antibiotic and pesticides use can result in the evolution of antibiotic resistance. Changes in weather patterns, like climate change, can make things like hantavirus and malaria show up in places it didn't. It means more vectors survive. There's more vectors, more chance to spread the disease. Malaria showing up in places malaria wasn't, because the mosquitoes aren't dying off. Modern transportation of diseases like the West Nile virus and cholera. Ecological disasters, war, expanding settlements in places that are in some parts of the world, like people cleared out parts of the African rainforest, and then they start getting Venezuelan hemorrhagic fever. There's animal control measures, like they used to spray for mosquitoes. So when the deer population goes up, there's an increase in Lyme disease because there's not enough predators. So maybe we need to have animal control measures like hunting. Public health failures. One of the things you guys probably don't know about. So back before Russia, the Russian Federation was the USSR, and which was multiple countries under Russia. Russia was part of the USSR, the United Soviet Republic, and Communist Republic of Russia, and all the countries it controlled. So it was massive. USSR we were in a Cold War with the USSR. When the USSR broke apart, the fall of that, and so those countries that we hear about today, like Ukraine and things like that, became their own countries again. These countries started becoming their own countries. Public health fell apart, and there was a diphtheria outbreak. Okay, so epidemiology, we always try to study when and where disease occur. That was the 1800s. Snow, a person named Snow, determined that cholera originated from a water pump in London. Semmelweis, puerperal fever during childbirth. That if you wash your hands then you're less likely to have problems with the women dying of puerperal fever. Nightingale, statistics to show that typhus, poor food, and unsanitary conditions were actually killing the soldiers, making them sick. Florence Nightingale, famous nurse. Okay. Descriptive epidemiology is collecting all the data that actually describe the occurrence of a disease. Who's infected, for example, Snow's research on cholera. Experimental epidemiology is looking at experiments to test a hypothesis about a disease, like Semmelweis on puerperal fever. Analytical epidemiology compare a disease group and a healthy group, like Florence Nightingale. Okay, these are all types of epidemiology. Last thing I really want to talk about is morbidity, mortality, things like that. Center for Disease Control collects, analyzes epidemiological information in the United States. The morbidity, mortality weekly report was out, the MMWR, and there's their website if you'd like to go to it. It's located in Atlanta, Georgia. The morbidity is the number of people affected by disease in the population. Mortality is the number of deaths. They're not the same thing. They are different. Okay? All right. So that is it, and I hope you enjoyed it, and I'll see you in the next one.